So I'll be talking today about expanding therapeutic modulation of brain circuits with a focus on my work in obesity and overeating. We tend to model overeating in mice with binge eating behavior, and so I often speak of overeating in the context of binge eating. Binge eating is perhaps the most severe form of overeating and very common in obesity as well as eating disorders. But obviously these behaviors are specific, and it's important to be rigorous when we discuss them. BITES is the name of my overeating suppression program that I brought to Penn from Stanford, and it's a cute title, but I do believe there's something very important about the timing of the bite, and you'll see exactly what I mean by that in a few moments here. John did teach me that if you don't have disclosures, you're doing something wrong. At this time, I'm not getting any consulting fees from ZAP. I would love to be, as another disclosure. But I am a consultant for Boston Scientific. I used to be one for NeuroPace, but because of my federal grant with them, I'm no longer a consultant with NeuroPace. This is the concept in schematic form of a treatment that I've envisioned since I was in college. I saw my first deep brain stimulation in the very room I'm doing deep brain stimulation now, performed by Gordon Baltuck, when I was a sophomore in college, and I never really wanted to do anything else. But I did want to apply it to novel indications, and I was always very interested in obesity and compulsive behavior, perhaps in the context of addiction. But I knew that we needed to study this behavior in person, in a laboratory setting, before allowing patients to receive this treatment in their homes. And studying addiction in a laboratory setting is a bit more challenging. Not impossible, but more challenging. So we started with obesity to try to not just stimulate and prevent behavior, but also to predict it, develop an algorithm to sense an electrical biomarker represented here on the right side of the screen that seems to correlate with the wanting of those donuts, and then delivering a intervention electrically to that same area to prevent the behavior. The rationale for this is obvious. Obesity is one of the most common public health crises in America, and over the past decade and a half has grown to epidemic proportions, affecting more than 30% of our country. This obviously is even more prevalent in our underrepresented minorities. And so in the early 2000s, I put a team of thought leaders together when I was at Penn, this was when I was in medical school, to brainstorm how to do deep brain stimulation for obesity. Given my interests in compulsive behavior and impulsivity, I was interested in the circuits that went awry in obesity, but also other types of compulsive behaviors that relate to addiction. And what I learned was that indeed the molecular and biochemical mechanisms of different kinds of reward like alcohol, opiates, stimulants, nicotine, they all converge on these dopaminergic neurons that project from the midbrain to the nucleus accumbens, and that includes food. And that's been extremely well studied, not just by me, but by many. And so one of my first studies when I was a resident at Penn was to test this out in a mouse. Some of the psychiatrists in the room might know John O'Riordan, who was a recent visiting professor at Penn, and I credited him with giving me the gumption as a neurosurgery resident to learn how to do deep brain stimulation in mice, to simply ask the question before going to humans, is this idea worth pursuing? Can we actually block overeating in a mouse with deep brain stimulation? And indeed, when mice are exposed to high-fat food, they develop binge-like behavior, and if we stimulate when that exposure is present, we can prevent them from eating that food. And interestingly, it didn't stop them from eating their standard chow, just high-fat food. And you can see that on day 0, which is the third figure on the bottom, there was a significant suppression in how much high-fat food they were eating. And when the device was shut off the next day, they went back to their normal binge eating habits. But when the device was turned back on, the behavior again was ameliorated, much like we would see with tremor. When the device is shut off, the tremor returns, and when the device comes back on, the tremor ameliorates. We carried this out for weeks at a time, not just 2 days, and demonstrated that this was reproducible and durable. But this wasn't continuous deep brain stimulation. We were just delivering stimulation when the high-fat food was present. So when I went to Stanford, this was my first faculty position. I had a K award, and I worked with Rob Malenka, who's sort of the father of addiction neuroscience. And also, I credit him with much of the discovery of optogenetics, as Carl Daesroth was a postdoc in Rob Malenka's lab when he discovered optogenetics. So Rob is not just a wonderful scientist, but also an amazing enabling mentor who doesn't get in people's way and actually just puts wind in their sails. In any case, what we learned in Rob's lab, which is a synaptic electrophysiology lab, is that there's a signal in the nucleus accumbens that seems to predict binge eating in mice. And you can see when mice are exposed to their standard food, what we call chow, there's very little activity as the mouse approaches the food right before their bite. When we expose them to high-fat food for the first time, they exhibit neophobia. They don't really want it. But when they take their first few bites, they realize that it's something that's very palatable. They also learn that we take it away from them, and that's how we provoke binge eating in mice. But after 10 days of exposure, you can see that the activity in the nucleus accumbens in the very same mice has changed, and we hypothesized that we could use this type of low-frequency signal to guide stimulation. I replicated my effects in blue of doing open-loop deep brain stimulation during exposure when I was at Stanford. We then also, in orange, manually stimulated these mice. This is, again, implanted in the nucleus accumbens. And we would sit in a room remotely, and as the mouse was approaching the high-fat food, my postdoc, who was Hemming Zwu at the time, would stimulate, and the mouse would walk away from the food. And then as the mouse would return to the food, he would stimulate again, and that was what we called manual stimulation. I tried to get that approved by the NIH, but self-stimulation was not something they were interested in, though I actually believe that would be a very reasonable therapy. What we then did, and you can see there in random, which is on the far right in black, what we then did was we calculated the amount of times we stimulated the mice during that 1 hour period. And we then just randomly stimulated them with the same amount of stimulation. And we had absolutely no effect on the behavior. When we programmed the device to stimulate in response to this low-frequency signal, which is delta band, increased power in the delta band, or a peak in delta activity, that's what you see there as RNS. Now I call this responsive deep brain stimulation, where we would actually stimulate only when the delta peak reached a certain threshold, and that seemed to mimic the effect of manual stimulation, but this was completely automated, and this was published in PNAS. This is a representative video of what we saw in our mice. This was actually the first mouse that we did this in as part of a pilot. And you can see here the food was delivered in the top left of the screen, and as the mouse approaches the food, in red, the stimulation gets turned on. And the mouse doesn't seem to have any adverse effects. It just seems to lose interest. When the stimulation gets shut off, however, he goes back to trying to eat again, but then it gets turned back on. So as being part of this amazing Malenka laboratory, which, by the way, there's no graduate students in Rob Malenka's lab. He has 15 to 20 postdocs, and these are some of the best scientists in the world. So for me, it was like a toy store of different types of manipulations that we could do to try to understand what we were doing here. There's a lot of different circuits here that we're modulating. The nucleus accumbens, like the subthalamic nucleus or the globus pallidus, is a deep brain structure that receives many different projections and projects itself to many regions of the brain. So it is a challenge to try to completely understand how this is working. And often when people say to me, well, how does deep brain stimulation work? I just think it's kind of a ridiculous question, because there's really no way to answer that with complete confidence. But we can scratch the surface on its mechanism, and that's what we tried to do here. What we did was, using optogenetics, we injected channel rhodopsin into the prefrontal cortex via a viral vector. and we allowed about a month period of time to allow that transfection to get to the terminals of the prefrontal cortex fibers that project to the nucleus accumbens, specifically to the nucleus accumbens shell subregion. And we know that interconnection is robust based on years of work in the mouse and other species. And what we found was that when we actually stimulated with light, again, to manipulate the synapse that was transfected with channel rhodopsin, we would actually decrease how much high-fat food these mice were eating. So what it seemed was that activating the prefrontal cortex terminals, not just the prefrontal cortex itself, but the terminals within the nucleus accumbens, that seemed to mimic the effect of doing deep brain stimulation to that area. So at the very least, I think we can say confidently that deep brain stimulation of the nucleus accumbens seems to be modulating the prefrontal cortex projections within that local area. So I wanted to tackle this in a human and examine this circuit better using imaging and to study this top-down frontal control circuit. And if we could better understand this circuit in a human and confirm some of our findings in mice, then perhaps we could better also understand how deep brain stimulation targeted to this area in a human would actually work. Because, understanding its mechanism in a mouse is a nice step forward, but it's not the human state. So some of you probably know Dr. Barboza in the back, who's my postdoc, now a senior research investigator in my laboratory and an absolute expert in tractography, joined my lab and co-mentored by an MRI physicist at Stanford. We were able to define the subregions of the nucleus accumbens and specifically used the prefrontal cortex or the ventral medial prefrontal cortex projections to the nucleus accumbens to delineate these subregions. And as we hypothesized, and this is also similar in the mouse brain, indeed the ventral medial prefrontal cortex has robust projections to the nucleus accumbens, specifically to the ventral medial subregion of the nucleus accumbens, which we define as the shell subregion. What was interesting, now teaming up with one of our eating disorder colleagues who had a cohort of binge eaters with this type of imaging, we could actually study the connectivity index between the prefrontal cortex and the nucleus accumbens, specifically, again, the ventral medial prefrontal cortex, the same region that we targeted in the mouse. And what we learned was, quite simply, that as the BMI increased, the connectivity index went down. And that is in line with the hypothesis that these types of individuals are impulsive. They have impaired top-down control. Teaming up with my career mentor, Murray Grossman, who is actually a neurologist at Penn, and one of his colleagues, Jim G., who developed some of the methods to understand to actually implement voxel-based morphometry, looking at cortical thickness. This is a controversial area in the imaging world, but nevertheless a method that we implemented here. And we indeed found that the gray matter of the ventral medial prefrontal cortex seems to thin as BMI increases. And not surprisingly, the thickness of the ventral medial prefrontal cortex and the connectivity index between the ventral medial prefrontal cortex and the nucleus accumbens also seem to directly correlate, suggesting to a certain extent that perhaps the connections between the top-down circuit here are impaired in these obese individuals, and the amount of perturbation there directly correlates to the BMI. Note, these are all patients that binge eat as well, so they're not just obese patients, but they're obese binge eaters. And this is not just a structural finding. Daniel also found that resting state connectivity between these two areas was also significantly decreased as the BMI increased. So the obese binge eaters exhibited significantly less functional connectivity in this circuit than lean individuals. So then going back to the mouse, I like to go back and forth. I luckily was able to implement a technique that I wanted to implement since the first days I was at Stanford, where we use calcium imaging. This is kind of like the reverse of optogenetics, using light fluorescence as a way to record from the brain, not just electrophysiologic recordings, but also doing pairing action potentials to fluorescence using viral vectors. And in a very similar way that we studied this circuit with optogenetics, we studied this circuit using fiber photometry. And this is calcium imaging, essentially, carried out again by Dr. Wu and Bina Kakusa, a phenomenal neurosurgery resident now at Stanford, one of my mentees when I was there. And in this study, we tackled implementing this technique, which was a challenge in and of itself, with deep brain stimulation. And we did deep brain stimulation continuously and deep brain stimulation responsively, like we were doing in the first PNAS study. And what we found, it's hard to see on the bottom left, but what we found was that there's a surge in calcium signal that lasts about 10 seconds when you do deep brain stimulation. But then the circuit's quiet. There's very little physiology that can be measured there with calcium signal, suggesting what we probably all assume, that deep brain stimulation does exhibit some sort of functional lesion to this area, though there is a brief amount of surge that might have something to do with the fact that when you turn these devices on, patients just feel something, but then they habituate to it. But when you do responsive deep brain stimulation, stimulating in response to that delta detection, what you can see is not just that surge in the beginning, but a surge that is intermittent and actually fairly reproducible. And if you think about turning a deep brain stimulation device on in an OCD patient or a depressed patient, they feel great when they're in your clinic, but then they go home and they're not feeling great anymore. I think we've all seen that, as those of us who have taken care of these kinds of patients. But perhaps if we stimulate them intermittently, the circuit doesn't habituate to that effect. And so we actually hypothesized, based on this finding, that we could rescue the circuit perturbation that Daniel defined in his obese binge eaters, because we could activate this circuit locally in the nucleus accumbens. So could we restore inhibitory control in these patients with obesity? So the hypothesis is this. So in the absence of binge eating, you have your somewhat normal circuit and connections between the ventral median prefrontal cortex and the nucleus accumbens. And by the way, it's a bidirectional circuit. It's not just top-down control, but it's also down-up. But let's oversimplify it for the case of simplicity. In obese binge eaters, there's this perturbation where these connections are perturbed, let's say impaired, although that might be an oversimplification as well. So the goal would be to try to rescue this circuit, like we did in mice, but in these human patients, to try to restore normal control. So we were granted what I consider an investment from the NIH in the form of a U award to study this in a first-in-human clinical trial. This is a representative patient. We've treated 2 patients to date. The rest of the study will be conducted at Penn, and we are about a week away from IRB approval to implant the remainder of the patients. But I'll share the data with you of the first 2 patients. I will admit none of this is published, but it is under review at Nature Medicine. So the first patient, and she's, I would say, completely representative of any patient that we're going to enroll, female, nearly 300 pounds. She failed gastric bypass surgery. She has binge eating disorder, exhibits loss of control eating multiple times a week. She's an emotional eater. She's tried everything to try to lose weight. Nothing has worked. We implanted her about a month before the pandemic started. Great timing. But obviously all researchers were impacted in a similar way. We used a device that was capable of sensing, by the way, which I'll talk about, and that actually allowed us to continue our research. So this is a paper that will be submitted in the near future to Neuron. I'm actually reviewing it on my flight here and my flight home. So again, unpublished data. I generally present on unpublished data all the time, to my peril. But I thought it was important to try to leverage what we've learned from Parkinson's. At least, when we started doing deep brain stimulation for Parkinson's, awake physiology was gold standard. Now we debate that, but at the very least, we don't know exactly where in the nucleus accumbens we want our electrodes to lie. And so we hypothesized that it would be important to try to do a petitive mapping, analogous to sensory motor mapping, which is standard of care for awake deep brain stimulation for Parkinson's or essential tremor. So we developed a task. I will say this was a combined effort between my lab, Nolan Williams' lab. Daniel was heavily involved in this to help ensure it was successful. Austin was a medical student now applying in neurosurgery, where we asked patients to take pictures of food that they loved. And we believed that personalized pictures were critical. And then we only included highly rated pictures, I believe 8 out of 10, 8 to 10 out of 10, I should say, representative of highly appetitive food stimuli. And then we also provided neutralized images, controlled for the color, but neutralized and not food related. And interestingly, what we found, and while this might not be statistically significant, this was a clinical assay. We weren't doing research in this way in the operating room. We were simply trying to ensure our electrodes were placed in a physiologically relevant part of the nucleus accumbens. And you can see in blue a robust change associated with pictures that were highly rated. And I'm going to show you a video of that. And this is all microelectrode recording. This is not local field potentials. This is all spike activity. And I want to credit Kai Miller, who is one of the smartest people that I know, a PhD, MD, PhD neurosurgeon with a PhD in physics and math, who recreated my operating room into a laboratory when he was my fellow at Stanford. And here you can see a brief audio file of what we heard in the operating room. I'm going to increase the volume here. It's already up. Okay, here we go. So the nucleus accumbens, people often say when they're doing deep brain stimulation for OCD, well, the nucleus accumbens is very quiet, so there's no value in physiology. And we don't believe that's true. Oh, sorry. Because we believe you have to provoke that activity. So you can see when this patient was shown a neutral picture, it was quiet. So I can understand why people don't do physiology in the nucleus accumbens, but when we showed her a picture of her homemade soup, there appeared to be some signal correlated to this appetitive food stimulus. This individual doesn't like sweet foods. She likes really salty foods. Mexican food was her most highly rated food picture. In any case, in addition to ensuring the physiology of our target, we also wanted to confirm that we could elevate their mood ever so briefly during moments when they were at risk, because most people that binge eat exhibit a pre-meal negative affect. They feel down, then they binge eat to make themselves feel up. So Nolan Williams and Ian Crowder came to the operating room. These are two interventional psychiatrists at Stanford, to modulate this area with the electrodes that we implanted and demonstrate that we were indeed able to elevate their mood significantly. One of the patients started laughing, grinning, things that we have seen in our patients with OCD and depression. And then we also obtained an intraoperative CAT scan to confirm that we've engaged the circuit of interest and targeted the very region of the nucleus accumbens that Daniel defined previously. So just a brief recap, and I think I have about 5 minutes, so I'll start rounding third base here. You got it. Surgical plan was safe, feasible, well tolerated in both of our patients. Both of our patients left the hospital post-op day 1. Seems that we were able to, for the first time, identify this appetitive unit that we're exploring further in this study. Test stimulation of this area induced a positive affect that we hypothesized based on our prior work, and we were able to use our intraoperative imaging to confirm target engagement. But now we have a device that we can use as a research tool because it can sense. This is actually NeuroPace's device that's typically used for epilepsy. We are using it here to do responsive deep brain stimulation, not cortical stimulation. Think of this like the epilepsy monitoring unit when you're trying to provoke seizures. This is an IRB-approved, consented, ethics evaluated and approved assay, and actually a fairly standardized maneuver in the eating disorder realm to provoke binges in the laboratory. Like seizures in the EMU, perhaps this isn't exactly what happens when these patients are at home, but it's an approximation. So this individual actually underwent a mood provocation with one of our psychiatrists, Deborah Safer. And then she was exposed to a buffet of her most preferred foods. This was after her breakfast and lunch, of which she ate all of. And she ate nearly 3,000 calories and reported feeling that this was a classic loss of control event. You can see that she is not just implanted with our device. You probably can't see the device, but believe me that there's a device in there. But she's also wearing an eye tracker, and she's wearing a monitor that allowed the neurostimulators raw data to be streamed in real time. So we could actually examine the moment of the bite and what was happening electrophysiologically at that time. So this is a very nice summary of what we found. Again, after a standard meal, she was provoked and started to binge in front of our very eyes. And we could use our own monitor through a one-way mirror, our own video camera, and an eye tracker to confirm that at the moment of the bite, like we saw in mice, on the left you see a standard meal where there's no activity, like we saw mice when they ate chow. There was really no food. You can see this on the bottom here. But when she lost control, the moment of the bite is time 0, there was an increased power in this low frequency range on this spectrogram. And this was very similar to what we saw in mice, something that we thought was really exciting. I've had 3 signal processors review this independently to confirm this finding is low frequency signals are subject or at risk for artifactual interference. And all 3 have found the exact same finding. So this was our candidate signal, confirmed it in this research laboratory. And both patients actually exhibited this type of signal as well. So perhaps the biomarker that we will use for loss of control will be standardized across this study. For now, we're planning on personalizing it, but for the first 2 patients, it was the same. In mice, this was in the low frequency range, up to 4 hertz. In humans, it seems to be both delta and theta. Nevertheless, this low frequency range seems to be important, much like it was in mice. Now, this neurostimulator also can do real-world ambulatory recordings, and I'm grateful for that for more than one reason, because during the pandemic, we were able to continue our research and continue our funding from the NIH. We paired magnet swiping, which is the way patients can timestamp in their electrographic trace when they feel at risk, or when they feel perfectly fine, or when they're about to go to sleep. This way we can actually do assessments of the electrical activity of this brain region during different times of the day, when they're at risk, not at risk for control assessments. And then we also had them take a food diary, which is the standard of care for eating disorder patients. Now, we can't know exactly when they took a bite in the ambulatory setting, although I'm interested in doing home video and wearable videos. That's a whole different discussion, but something that I'm exploring for a follow-up study that Daniel and I will be submitting this June. But what we found, and this is kind of interesting. Oh, that's my alarm. I heard John say that we were going to stay on time today, so I wanted to make sure I did. So I'll wrap up here in a moment. What we found was that the low-frequency signal in the nucleus accumbens bilaterally in both patients exhibited this increased power in this low-frequency range when patients felt like they were craving. So we used craving as a proxy for loss of control, something that most patients that have binge eating disorder share. And because of the time frame that the NeuroPace data is shared with us, we could look back about 1 hour. And what we found was that during that 1 hour period, there was increased power on average in this delta range compared to when people were just eating normally or at baseline. So while in the mouse and in the laboratory, it seems that this episodic biomarker is present moments before a bite, it does seem like it ramps up over time. And a more thorough analysis suggests that perhaps it ramps up starting about 30 minutes before the bite. Nevertheless, in the ambulatory setting, it's hard to know exactly when that bite was, but we've asked all these patients to swipe over their magnet right before they start their binge meal. Just a brief summary of our outcomes to date. Again, this is unpublished, but under review now at Nature Medicine. In our first patient, you can see, first off, the yellow dots represent her weight in pounds. But the most important signal here to look at is the blue trace. This is the frequency of loss of control. And this is actually our primary endpoint. And you can see when we activated the lead on the left, she didn't really have much of a response. And we didn't really expect it. We started at a pretty low dose. But of course, you know, I got a little nervous because I like to see immediate effects, but we didn't have one. And interestingly, it didn't really change until we activated the right side. Okay, so we needed to do bilateral stimulation. And obviously, since then, she's lost some weight as well, about 30 pounds.
To be clear, where you're stimulating where again?
Well, thank you for asking that. I'll actually show you that in a moment. But the location of the stimulation is the nucleus accumbens. However, it's likely that the volume of tissue that's activated proceeds out of the nucleus accumbens. And we are doing bipolar stimulation, so we're using 2 contacts. So it's pretty much the entire span of the nucleus accumbens. It's hard to be specific to just the ventral medial region. But interestingly, if you recall, when we turned her device on, we turned her on the left side. And the left electrode, you can see it on the bottom right, and you might see it better in this image here. The left electrode is not sitting in Daniel's streamlines. These are the streamlines coming from the ventral medial prefrontal cortex. And the reason they're not is because there was a blood vessel near her caudate. And so I had to implant this lead a little bit more lateral than I wanted to, knowing, this was 2 millimeters, knowing that we could likely engage that circuit with a higher dose of stimulation. But we didn't start with a high dose of stimulation. And we believe that this explains why it took bilateral stimulation in this patient to exhibit a robust response. In our second patient, again, you can follow her outcome here in blue and in yellow. Those are the 2 most important, the blue being the frequency of loss of control and the yellow being her weight. She's had a more robust effect. By the way, the first patient met the primary endpoint for the study, which allowed us to get approval to do the rest of the study at Penn. But in this patient, when we activated the left side, she had nearly an immediate improvement in loss of control. It wasn't something that she reported to us in the clinic, but when she went home, like a week later, she was like, gosh, this week has been life-changing. I don't crave peanut butter anymore. I'm just a different person. And that effect has been fairly stable since we turned it on, but got more robust when we turned on the right side. And you can see here, both the right and the left lead are sitting within Daniel's streamlines here. We believe that is why it's so critical to use tractography, at least for this target, using our methods to ensure the best result. But early days, only 2 patients, and these are our outcomes to date. Both patients have exhibited robust improvements in loss of control ED frequency, severity. This is the feature of all binges, loss of control. They both have decreased in their weight and body mass index as well. So we're very excited about this. And just some patient testimonials to close up. I'm noticing I'm eating less, being satisfied with less, smaller portions, better choices. Just thought I should share. Better choices, really important finding. I've had a couple bouts of lightheadedness while sitting in a chair, similar to the feeling of standing up too fast, but not as intense. They seem to be going away. So there were some potential mild side effects. This was right after her surgery. I haven't had peanut butter in 2 days. I've just had no desire for it. Could be all in my head, pun intended, and just don't want it, which is a kind of scary thing because I would eat it 3 to 4 times a day before. And our other patient said, if this is how my journey is going to be, I have a feeling it will be successful. I am so grateful to these patients for trusting us to take care of them and to partake in this highly unusual study. So I'll close with this slide. So what's the future? Is it radiomodulation, bringing it back to why we're all here? Maybe. So the issue with that is we're still trying to figure out where we're stimulating. And we have a device that has substantial spatial resolution issues. That is not a criticism. That's the only way to study this circuit right now with a closed-loop device. But lots of improvement for the future. And I think if we're going to use radiomodulation for a target like this, we have to know where in the nucleus accumbens we need to do it. There's other obstacles. So the nucleus accumbens is heterogeneous, and there's two major subregions. We believe the ventral medial subregion is critical. We don't know if targeting that area with modulation is sufficient because our stimulation fields likely traverse outside of this area as well. We don't know exactly where the delta biomarker is coming from. We're doing differential recording. That's bipolar recordings. And we can probably record from a couple millimeters away. So where is this delta signal coming from? And right now we have to detect it in the nucleus accumbens. So that could be a limitation for radiomodulation as well. I have ideas as to how to make that actually possible, but nevertheless, an obstacle. Is this the sweet spot? There might be another sweet spot. There might be something better. There might be something cortical to go after. Maybe we should be stimulating the ventral medial prefrontal cortex. It might be an easier target, but a much bigger one. That might not be a problem for radiomodulation, but it would be a problem for invasive stimulation. And the most challenging obstacle, I think, for radiomodulation for this is that the benefit seems to be in the timing. Chronic stimulation, based on our findings, seems to actually be less effective and also not behaviorally specific. In our mouse studies, we interrupted normal social interaction by doing continuous stimulation. There have been clinically meaningful, significant side effects of doing continuous stimulation to this area in similar patient population published recently by one of our speakers. So I am concerned about doing continuous stimulation in this, for this type of indication. And I have some ideas about how radiomodulation could be done episodically, but that's not necessarily what it would be best at doing. So lots of things to consider here. Lots of people to thank, most of whom are at Stanford, but I don't want to go over time too much. Brett, you went a little shorter than planned, so I appreciate the extra time. And if we don't have time for questions, hopefully we have some time to discuss at dinner.